As a last example, let's see how the GRN modeler app can handle cell loans. For that, we will use one of the archetypes of spatial patterning, the Turing pattern. Let's load from the example folder turing.mat. We can see that in this gene regulatory network, we have two nodes, node 1 activating node 2 and itself, and node 2 repressing node 1 and itself. To create this topology in which a node regulates itself, a new type called mix-in has been created. Also, if we look at the regulator properties box set, we can see that n, the Hill coefficient for the regulator, has been set as 6 to ensure high nonlinearity, which is a requirement for these Turing patterns. If we run a simple simulation, we see that the system oscillates a bit and then reaches a steady state. This is very important in case we want to simulate spatial patterns, because if this was not the case, our cell loan will continue to oscillate over time and we will not reach a stationary pattern. So once we've checked for this, we can finally run our spatial simulation. In this case, we leave loan of cells selected, so the colony properties box is inactive, and also the small boxes diffuses with colony cannot be modified because there's no colony in this case. What we can change instead is the diffusion coefficient, and in particular for this Turing pattern, it is important that the diffusion coefficient of the activator, Pn1, is low, so the activator diffuses slowly, and the repressor, Pn2, diffuses fast. So we can set this one as at 10 and Pn1 at 1. I will also reduce the number of time steps here, and we need to make sure that noise is higher than 0. Why is that? Because if our loan of cell was perfectly homogeneous, there will be no instability and no Turing pattern arising. While instead, if we make sure that noise is a little bit higher than 0, the small differences in the initial condition will lead to the formation of Turing pattern. Okay, so we're ready. We can run our simulation. Once again, we get plots as the system evolves. Once again, we have cell concentration. No, in this case, we're observing graphs for both node 1 and node 2 protein product. And you see that the final result, once the system reaches stationary state, is a dotted pattern, typical of these Turing systems. As always, if you want, you can export, save and export the figures that you have generated with the spatial simulation feature. And very importantly, if you want to create your own images and graphs, you can also save the data of the simulation using this Save Data button, which only becomes active after a simulation has been run. Okay, that's all. We hope this spatial simulation feature can be helpful for you to figure out whether your gene regulatory network have potential for spatial pattern, both at the colony level and at the cell loan conformation.